Hi, welcome everyone. In today's lecture, we're going to be talking about transplantation immunology, which is a pretty fascinating chapter. And what I really like about it is that it incorporates a lot of the same principles and fundamentals that we learned in previous chapters, simply in new contexts and applications. As we're going to learn, it's pretty T cell centric, so it certainly builds upon the activation and the effector functions of both CD8 and CD4 plus cells, with also a limited extent of B cells as well. And then certainly a lot from what we learned in our tolerance chapter as well. So before we start, I'd like to go a little bit more into the scope and the need for transplantation procedures in general, as I think this is really one of the applications where we start going from pure immunology and we really start seeing applications in engineering, specifically biomedical engineering and tissue engineering. So for instance, in 2019, there are still around 113,000 plus patients on the transplant waiting list. These are patients waiting for a viable organ to open up because they are in need. Now, unfortunately, last year, there are only above 36,000 transplants that were performed. So it really highlights the disparity between those in need and what the market or the available population has been able to provide. To highlight that perhaps a little bit more, in the central figure, we have a graph that shows the number of people on both the transplant waiting list, the number of transplants occurred, and the number of available donors as a function per year. And we see the sharp increase of those on the waiting list compared to the available donors. So we can definitely see the sharp rise in those on the waiting list is just getting steeper and steeper, while the number of donors is just not able to meet up that particular demand. To highlight that perhaps even further, every 10 minutes someone is added on the national transplant waiting list. Yet every day 20 people died just waiting for an organ to come up that never did. Of those transplantations that did occur, a vast majority, 83% of them, were kidney transplants, followed by a very distant second, 11% were liver transplants. And then there are very small percentages of heart, lung, and other organs that were transplanted. So hopefully this indicates the real need to figure out not only viable methods for transplanting donor tissues, but perhaps us in the future as biomedical engineers to develop natural and synthetically based organs, or even develop tissue and regenerative engineering methods that can fulfill this unmet need. Now, before we begin this chapter, I'd like to go over some terminology that we're going to use throughout this presentation. A graft in general is whatever is being transplanted. It could be individual cells or a collection of cells, tissues, or whole organs. We're just going to use the generic term graft for that. Transfusion is a particular kind of graft that we usually reserve for circulating blood, which does include cells, or even just the plasma without the cells. Those that are providing the particular graft would be the donor, and then the recipient or host is the individual that receives the graft. Now, because the reactions against the type of graft that you get are going to vary widely depending on the source that it's from, we have a few terms that are defined the types of grafts that you might get. For instance, autologous grafts or an autograft is a graft from that same individual. So this would be common in, for instance, a ligament replacement surgery where you take a piece of a tendon from maybe your hamstring to use it to repair an ACL or an MCL injury. A syngeneic graft is one transplanted from genetically identical individuals. This could be inbred animals, or it could even be identical twin in humans. Allogenic grafts or allografts are from genetically different individuals of the same species, and this is going to be the primary focus of this presentation. These are exactly the same type of grafts that we were talking about on the previous slide too. So a donor donates a graft, an organ, and then a recipient, a host on the waiting list receives that graft. They're from the same species, but genetically different. In contrast, a xenogenic or a xenograft is a graft that goes across different species, which is done very rarely to fulfill medical needs. And I see it most commonly in terms of research. So for instance, a xenograft tumor is a very common procedure in cancer research where a lab mouse or rat will receive tumor cells from a different host in order to grow a tumor, and then we can test chemotherapeutics or novel cancer medications on them. Now, those same prefixes that were applied to the word graft are also going to be applied to words like antigens and reactions in order to note where those particular antigens come from, specifically from the donor, from the host. Is it a genetically dissimilar but from the same species? Is it an antigen from a different species? So an alloantigen is, again, just like on the previous slide, an antigen from the donor tissue that's being reacted against the host immune system. And now for a super quick history and a half slide introduction to the general principles of transplantation immunology. So the technical hurdles of these transplant surgeries were historically the very first barrier and represent a significant breakthrough in medical technology. Now we consider many of these relatively straightforward for those trained in it, of course. And it's really after transplantation, battling and controlling the immune response that is now a major barrier. So controlling the immune reaction is really key to the survival of a transplanted organ and, of course, the host recipient of that donor tissue. In general, allogenic and xenogenic transplants invariably lead to rejection of a graft due to an adaptive immune response. So this is going to be dominated primarily by T cells, actually. We're going to find out B cells to a limited extent. But the innate immune response does have a role in this. It certainly does amplify the adaptive immune response. We've learned in previous chapters the synergistic relationship between the two. And then primarily, certainly due the surgical action of transplanting grafts, we see a lot of damage-associated molecular patterns due to the ischemic damage from surgery. And then we're also going to find out that natural killer cells themselves play a large role in this as well. So in order to get into it, I'm going to dive into sort of the evolution of our historical understanding of how the adaptive immune response came into play through a series of experiments in mice. Early studies show that whenever skin grafts from one particular inbred strain of mice was transplanted into a recipient of a different strain, so this would be genetically dissimilar, an allograft, the transplanted tissue was fine for the first number of days, even up to a week. We saw no sign of an immune response, but certainly after 10 to 14 days, we saw this rejection occur and the body would reject the transplanted graft. So this kind of gives us our first clue, just based on the time scale alone, that this isn't an innate immune response, but something more characteristic of the time it takes to develop an adaptive immune response. The second series of experiments would repeat this in the same mice, in the same recipients, the same hosts that received the first transplant. They were given a second transplant from that same original donor strain. And this time, the second set rejection occurred much faster, on the order of 3 to 7 days, in contrast to the original transplant, where it took 10 to 14 days. So this was the second clue that not only does this take a while to develop, but second set rejection has characteristics of memory in that it comes back not only more rigorous, but certainly faster. The third clue, and the real nail in the coffin, is when they repeated this experiment with a brand new set of strain B mice. Those had never received a transplant before. They would take the graft from the same donor, from strain A mice, put it in this new set of strain B, the first time they'd ever received this transplant, while simultaneously infusing these strain B mice with activated and memory lymphocytes from the second and first set of experiment mice that had already received this graft previously. And they were able to accelerate that second set rejection kinetics. They're able to achieve those, even though these mice have never seen these grafts before. And it was because they had these new memory and effector T lymphocytes from the previous strain that imparted this accelerated kinetics, similar to a second set rejection. Now, at the time, they didn't know exactly what they were infusing, but they did confirm later, scientists in general, that these were, in fact, T lymphocytes and B lymphocytes that would help impart these second set kinetics and ultimately confirm that this was an adaptive immune response that was ultimately responsible for tissue rejection. Now I'm going to bring you through 4 basic rules of transplantation using the same series of mice experiments. The first key principle is that a graft from a genetically identical inbred strain or a genetically identical person from one donor to a genetically similar host is generally never going to result in a rejection. So a syngeneic graft is pretty much the same as an autograft. And we know that autografts work because we ourselves have developed central and peripheral tolerance for our own antigens. So if we provide our own grafts to another host that's genetically similar, they've already developed the same type of central tolerances for our own antigens and therefore aren't going to reject these grafts. In contrast, an allograft from genetically dissimilar individuals or strains of the same species, or even outside the same species, xenograft, is invariably going to lead to a rejection of that graft. Now, where it gets a little bit more complicated is when we look at parents and offspring of 2 genetically different strains, or between, of course, genetically different people that aren't inbred. The first key principle is that offspring from 2 genetically dissimilar strains can successfully receive grafts and not reject them. This means that a parent can always provide a graft to an offspring because that offspring has developed tolerance to the antigens of both parents. However, you can't go the reverse direction because the graft from an offspring will have antigens from both parents. So if you try and provide a graft into a recipient that only has one of those antigens, for instance, the parent of strain A, they'll have to react against and reject any antigens from strain B that is from the other parent. So that means parents can always provide grafts towards their offspring, but offspring cannot provide grafts to their parents. Now, in truth and practice, we do this actually all the time, but we need a lot of immunosuppressants and other ways to combat and control the immune response. So this is only in uncontrolled situations. What will either lead to a rejection or a successful graft transplantation relies on genetic similarities or dissimilarities between the host and the donor. This does bring up a few key principles. One is that these antigens are polymorphic, which means that even individuals in the same species, what determines graft success or rejection is different amongst genetically different individuals. This is important because there's a lot of things in humans that actually aren't polymorphic. So despite our dissimilarities, I suppose, we're actually very similar biologically in a lot of ways, but it's the fact that these grain antigens are some of the few things that are polymorphic that make graft transplantation so difficult. Also, the rules between offspring and the parents bring up the fact that these graft antigens are codominant, meaning that every individual inherits these genes from both parents and both alleles are expressed in various amounts. So that means the offspring will always have these antigens from both parents and they're always involved in rejection. It's not like some are dominant and others are suppressed. In these cases, these graft antigens are codominant. Now, if you're wondering what these graft antigens are, you've heard about them many times. These are the major histocompatibility complexes, also synonymous with human leukocyte antigens, so those terms are used synonymously. The MHC complexes refer to both the genes that encode these grafts and also the proteins that themselves are transcribed as a result of those genes. So most commonly in previous chapters, we've talked about the complexes, the proteins that hold both self and foreign peptide antigens on the outside of cells. They're also the responsible genes that encode those as well, so they share the same name. Now, if you ever wonder why they're called major histocompatibility complexes, it's also because there are minor histocompatibility complexes that are also involved in graft rejection. We'll talk about this a little bit later. As their name suggests, the major ones are responsible for the strong and rapid rejection reactions, and the minor ones are responsible for weaker and slower responses. Now that we finally brought the topic back to MHCs, we can bring it back to T cells and the recognition and presentation of antigen or these MHCs back to T cells. Now, in previous chapters, we really only talked about the MHC as a means of carrying whatever antigen that gets recognized by the T cell. What I really want to stress here is that the MHC is going to be the primary focus of this chapter, and it's absolutely 100% itself responsible for graft rejection or acceptance, not the antigen. Okay, so what's polymorphic between people are the MHCs. It has nothing to do with the antigens that they support. And what I mean by that is that all the cells that we have, they're even across different individuals that aren't genetically similar, they're incredibly similar. Okay, so all of the leukocytes that we learned about, even other tissue cells, think of the lipid membranes, the proteins inside. Yeah, maybe the DNA inside is different amongst individuals, but that's way too variable to either code for specificity in our lymphocytes or even code for tolerance as well. So I want you to start just thinking to yourself, the only thing that's different is going to be the MHC. So that's what is either going to trigger activation or tolerance in these host or donor lymphocytes. So now that I stressed that, whenever we have a graft tissue in a new host, there's going to be two ways that host T cells can recognize that intact graft donor MHC. The first one is through what's actually a little bit more complicated than it seems, is direct recognition or direct presentation of those MHCs. This is where the donor APCs, think of it as either dendritic cells initially or even other APCs after the fact, have intact MHCs that are themselves compatible with our own lymphocytes. So they're displaying either self or foreign peptides, meaning self peptides that they picked up from the donor, or somehow later they picked up foreign peptides from the recipient. They are themselves presenting these MHC complexes to now the host, and they create an alloreaction in that the host CD4 cells or the CD8 cells can directly bind to those MHCs, get activated, and then the effector versions will carry out their mechanisms in response to seeing those semi-MHCs now that they're activated. Now, that itself sounds pretty straightforward, but it can get a little bit tricky on how these T cells recognize either the MHCs or the self-reformed peptides that are on the MHC. So first, let's go over the diagram on the right, which covers how a normal T cell receptor interacts with a self-MHC carrying a foreign peptide. And here we see those docking sites, plus the unique configurations, that particular foreign peptide that somehow fits in the keyhole of the T cell receptor, if I'll use that analogy. And here it's the foreign antigen that provides 100% of the specificity to that T cell receptor. Now, in order for allorecognition to occur, a host T cell has to somehow be able to recognize an allogenic MHC, which it's not designed to do. So it can do this in two possible scenarios. One is where it's partially specific to the MHC, but then a self-antigen, meaning one that's being carried by the allogenic MHC, kind of fulfills the remaining sort of slots that provide that T cell specificity. So in here in this diagram, you can see that one of the middle keys of the allogenic MHC fulfills one of those specific slots while then being complemented by the self-peptide. Now, again, what I want to stress is that there's not that many polymorphic self-peptides that this antigen-presenting cell could be presenting. So it's highly unlikely that the T cell will be specific against that self-peptide, which is why it would need some of the uniqueness of the MHC in order to be something specific that T cell recognizes. In the second possible scenario, we see here T cells that are specific for only the MHC, and that self-peptide is simply a means for that MHC to be built and to be held together. There is no interaction with the T cell and that self-peptide. It's entirely specific to the MHC. So again, I can't stress enough how unlikely it is for that self-peptide to be reactive against that T cells. Again, humans simply don't have enough polymorphic peptides that aren't related to the MHC to be displaying in order for this to happen, direct recognition. Now, as a result of direct recognition, since CTLs, or effector CD8 cells, can't directly recognize these, they can immediately and directly carry out their cell-mediated cytotoxicity function. This is the formation of the immune synapse, the release of granzymes and peripherum that directly lead to the killing of that cell. What's nice and direct about this process is that there is no need for the MHCs to be processed by host APCs. Donor APCs are entirely responsible for the initial activation, and they're the ones that ultimately, again, get killed by the host CTLs because they can recognize those MHCs directly. So this kind of raises the question how T cells could sort of naturally evolve to recognize these MHCs. A likely response is that there's some sort of natural affinity between MHCs and T cell receptors in general. And certainly we also know about the positive selection process, how positive selection promotes the survival of cells that at least have some affinity to an MHC. And of course, these MHCs are carrying self-antigens. So they certainly promote all T cells that at least have a weak affinity, which certainly opens the possibility that some of those have a strong affinity to allogenic MHCs. These are MHCs that they never would have seen before. And of course, that brings up negative selection, which would only eliminate T cells with a strong affinity to self-MHCs. There is no elimination process for a negative selection of allogenic MHCs. There are no allogenic MHCs in the central lymphatic organs. So that's only a possible mechanism of how these T cells evolve to have just this byproduct affinity of foreign or allogenic MHCs. Now, a typical response from a direct recognition of MHCs is an incredibly strong immune response, much stronger than we would find against any particular foreign pathogen. And this is for a number of different reasons, but realize that MHCs are themselves very generic structures. So 1 to 10% of all host T cells will react against allogenic MHCs, whereas normally 1 in every 10 to the 5th, 10 to the 6th power T cells are specific to any single antigen. Also realize that these donor MHC cells can present a variety and many different types of peptides. Actually, I didn't know this, but microbes, bacteria, viruses themselves have relatively few peptides that can be presented, whereas we have tons of peptides. And not only that, peptides are simply a means to carry the MHC in this case, as opposed to we used to think vice versa in the case of when we were looking at foreign antigens. In this case, the self or foreign antigen is, we don't really care about that at all. It's the MHCs that are themselves very generic. They can carry anything, and these T cells will react to the MHC Again, we're not thinking of the T cells reacting against the antigen in this particular chapter. And for that matter, these allogenic MHCs can present both foreign and self-host peptides. It doesn't matter at all. They're still displaying those MHCs. And lastly, there are memory T cells that respond to allogenic MHCs for the same reason, even though they might have been developed through different means, or maybe they came about through previous transfusions, transplantations, even multiple pregnancies can induce this phenomena. We have memory T cells that are latent in tissues that will react really strongly, faster than other sort of naive T cells that have to be presented. So those are a number of reasons why direct recognition of MHCs results in a very strong reaction. Now, as opposed to direct recognition by T cells, there's also indirect allo-recognition by T cells, or indirect presentation. So this is very similar to what we think of in terms of foreign antigens in foreign bodies. This is where our own host antigen-presenting cells process donor cells that have themselves their MHCs. We produce peptides out of these allogenic MHCs. So then the phagocytic pathway present those antigens on both class 2 MHCs. Through cross-presentation, these dendritic cells will present them on class 1 MHCs. This activates both CD4 plus and CD8 plus cells to produce those effector and memory T cells. The CD4 plus themselves go on to react with B cells to produce antibodies, high affinity antibodies, class switching, all that. Those CTLs and CD4 cells, however, can only produce their effector functions through cytokine-mediated pathways. This is, of course, the nature of CD4 plus helper T cells. However, the CTLs, this is sort of them not being able to do the direct job that they're assigned to. They're expecting to find class 1 MHCs of infected cells with viruses or cancer cells. However, unlike those CTLs that can recognize through direct presentation, these CTLs are not able to do that. They're somehow not compatible. But about 90% of the T cells are going to be like this. They're not compatible with the donor MHC. So they're never going to be able to directly recognize those MHCs. Thus, they only have the option of using their cytokine-mediated functions. They do release IF and gamma, just like Th1 CD4 plus cells. They produce inflammatory reactions that augment the innate immune response and also trigger macrophages and neutrophils. So those CTLs can only kind of do their secondary job and not carry out their direct cell-mediated cytotoxicity jobs. And then lastly, B cells contribute to humoral mediated toxicity through antibody-mediated cell toxicity. So these antibodies will activate complement, they'll activate natural killer cells, they'll activate phagocytic receptors on other cells of the innate immune response. So I kind of like this slide. It's a very nice overview of all the effector functions in many chapters in about 30 seconds. So nice. And you know what? If that half slide wasn't enough, maybe I'll do it for just another full slide and talk about the activation effector function of these lymphocytes. So first, how do these APCs actually present their antigens? So after the surgically placed graft is put into place, it can reconnect and repair those lymph ducts that can then allow travel of donor antigen presenting cells, dendritic cells, to travel to the host lymph nodes. So these donor APCs will mingle in with host APCs and all the host T cells. The host dendritic cells will also carry out their donor antigens, migrate to the side of the graft and migrate back. So again, there's a mix of both host and donor dendritic cells presenting these antigens and triggering the activation of T cells. Resident memory T cells of the host will react at the site's local immediately without the need of these dendritic cells because they're already primed and ready to go. Now, of course, the reaction of naive alloreactive T cells does require co-stimulators. This is exactly the same as naive T cells that react against foreign antigens. You need the co-stimulators on dendritic cells in order for those initial activations. It also is somewhat important even for memory T cells. So let's not think of it binary. Co-stimulators are always into play, just less so with memory T cells. So in this case, because there's no foreign antigens per se, the way we usually think about them, co-stimulators are usually brought about due to the ischemic damage of the surgery itself. So let's think of the damage-associated molecular patterns caused itself by the transplantation procedure that cause for the stimulation of things like toll-like receptors and other molecular receptors that react to damage-associated molecular patterns instead of foreign-associated molecular patterns. Therefore, a very effective strategy in order to promote the success of graft transplants is to block co-stimulators in order to prevent this from happening. Now, as far as the effector functions go, I really want to repeat this a few times. CTLs, those effector CD8 cells, can only carry out their cell-mediated cytotoxicity pattern when they're activated through direct recognition. So this not only achieves their activation and their priming, but also whenever they go to interact with the cells, they have to be able to interact with those MHCs specific to the donor. Okay, usually they can target and recognize their own host ones, and it's only when they're compatible with the donor MHCs can they carry out these cell-mediated functions. Okay, so this requires intact allogenic MHCs, not ones processed by the own host on their dendritic cells. But also these CTLs can also carry out their secondary functions, which is to release inflammatory cytokines like IFN gamma. Those CD8 cells that are activated through indirect recognition will not bind to these peptides. They cannot carry out the cell-mediated cytotoxicity because they are not compatible with those donor MHCs. So they can only be effective through their cytokine-mediated pathways. Again, this is the release of IFN gamma and other cytokines. Now, CD4 plus cells can be activated through both direct or indirect patterns. It doesn't matter at all. But regardless, they carry out their functions through cytokines. So they don't care at all whether or not they're compatible. They just care whether or not they get activated at all, and they're still going to release their cytokines. So that doesn't matter at all for CD4 plus helper T cells. Now, as we learned in previous chapters, CD4 plus cells do activate B cells, and they help produce high affinity antibodies that are a class switch to primarily do IgG antibodies. And these antibodies will react against peptide sequences on both class 1 and class 2 MHCs. So that means that the B cells are themselves specific to peptide sequences on those MHCs, on those allogenic MHCs to be specific. Now, what this does is that the antibodies activate complement. They mediate functions of neutrophils and macrophages through Fc gamma receptors. Same thing with natural killer cells, having those Fc gamma receptors as well. Now, unlike foreign antigens, where we learned that a particular microbe might elicit a more dominant response by one particular aspect of both the innate and adaptive immune response, for instance, viruses we associate with CTLs along with cancer, extracellular pathogen with antibodies, intracellular bacteria with phagocytes. It's different for graft recognition in that all these responses pretty much occur simultaneously. Again, remember that MHCs are themselves very generic. So you're always going to have a lot of antibodies that can react to them, different CTLs that might be programmed to react to different foreign antigens, hypothetically, but they all end up reacting against the same allogenic MHCs. So you always have a mix of B cells, CD4 plus cells, and CD8 cells that react against the graft. Because we are dealing with MHCs, CD4 plus and CD8 cells do come into the mix. But again, because these peptides are processed, B cells can recognize peptides. B cells come into the play as well, as does allogenic or alloreactive antibodies. Now, despite that, some of these reactions do occur at different times. Because of that, historically, patterns of allograft rejection have been divided into 3 subcategories based on their timeline, and these different timelines or patterns tend to have their own mechanism and their own pathological consequences. So let's go over those. The first one is what we call a hyperacute rejection of a particular graft. And as its name implies, hyperacute, it actually occurs on the order of even minutes to sometimes hours. And it's mediated by pre-existing antibodies. And the pathological consequence tends to be intravascular thrombosis, so a lot of clotting. Now, what causes this is that these antibodies will start to activate complement that itself leads into a cascading reaction that eventually promotes both the thrombotic mechanisms innate in our bodies and also disrupt our own anticoagulation defenses. Now, historically, these are due to natural IgM antibodies that react against the various different types of blood types that we might not be compatible with. These are called the ABO carbohydrate antigens that are present on blood cells. So this is why thrombosis is such a key to this. We've now been able to successfully screen for compatible ABO types for quite some time, so we've all but eliminated this problem. Now it's the result of IgG antibodies that are not themselves natural. If you need a hint of that, it's because these are IgG, so we think, okay, this must involve T cell help at some point to have undergone this class switching from what used to be IgM to now IgG specific antibodies that are specific to different types of protein alloantigens that are different from these ABO blood type antigens. So these are general results of previous exposure to these alloantigens, maybe through blood transfusions, other previous transplantations, or even multiple pregnancies can bring this up as well. So many people have these IgG antibodies just now as a result of these previous inductions just in them and waiting to pounce on a particular graft that might be transplanted. Following the hyperacute phase, we go into the just acute rejection phase, which is on the order of several days to even weeks. And then possibly, if we were able to stave that off with some immunosuppressants, this can actually go into months and even years as well. So this one is generically mediated by both T cells and antibodies, and it leads to killing of the graft parenchymal and endothelial cells. Parenchymal, by the way, just means the cells that have their own unique functions to that particular graft, that organ. And then endothelial cells we commonly associate with the endothelial lining of blood vessels. So the mechanism can be divided into both cellular and antibody-mediated mechanisms. So this, of course, would be the cell TL-mediated killing of direct graft cells, also indirectly through their secreted cytokines, as is the same with the CD4 plus cells that mediate their functions always by secreted cytokines that activate the innate immune leukocytes and inflame cells as well. The antibodies also bind to MHCs on these vascular endothelial cells, activate complement, and then pair up with cells of the innate immune responses, just like natural killer cells and phagocytes that mediate their functions through the Fc portions of those antibodies. After the acute phase, this invariably leads us to the chronic rejection phase, which can go from months to years, or just generically ongoing throughout the lifetime of the patient. This is mediated by the alloreactive T cells primarily, less so antibodies in this case. And the classic pattern for chronic rejection is arterial, meaning blood vasculature occlusion, via the proliferation of the smooth muscle cells. They eventually just keep growing and growing. You get these lesions that eventually just block these vessels because they've grown so much underneath the endothelial lining of the blood vessels. Since we haven't talked too much about how cells of the innate and adaptive immune response interact with non-immune cells, I'm not going to go into this in too much detail. So it's just suffice it to say that the T cells secrete IFN gamma and other cytokines that do stimulate the proliferation of these vascular smooth muscles that themselves compromise blood flow through this mechanism. Now, with modern day immunosuppressive therapies, we've all but practically eliminated both the hyperacute and the acute phase. And it's really the chronic rejection phase that is the major hurdle in immune transplantation. So we've successfully brought a 1-year survival of kidney allografts to having a survival rate of over 90%, whereas the 10-year survival of kidney allografts has remained pretty stagnant for the past 20, 30 years at around 60%. So that's something that's been a big hurdle for us to improve. So a few times now I've vaguely mentioned the idea of using immunosuppressive strategies in order to increase the chances of graft successes. So now we're going to dedicate almost the remainder of a chapter talking about ways of preventing rejection. First up are a couple different ways that we can increase the likelihood of success for a graft transplant without even modifying or controlling the immune response. It's really more preparatory roles that we can increase the chance or likelihood that a graft will succeed. The first option is that when possible, we would like to take grafts from living donors as opposed to cadaveric or cadavers. So there's a couple different ways that this is possible. So certainly there are certain organs that are easily transplanted even in living donors. Things like the kidneys, half of a lung, like a lung lobe, parts of the kidney, pancreas, intestines. In those, we don't need to sacrifice the donor in order to have a successful graft. Now, of course, this is impossible with vital organs like the heart, and even in non-vital organs, just due to the sheer limitations of the available amount of organ donors or available organs. Sometimes patients just have to take what we can get. But we know some of the reasons why this is the case, why we see higher success rates from organs that are transplanted from living donors as opposed to cadaveric donors, is usually due to the trauma associated with whatever caused that person to die. You know, whether it's they're not technically dead, but maybe they're brain dead and they're brought into a hospital. Even organs from those types of patients where they're harvested on a technically still living donor have reduced success rates compared to those from living donors. And we can certainly hypothesize that that trauma will induce a lot of damage associated molecular patterns and a lot of cytokines present in the donor tissue that would thus help stimulate the adaptive and innate immune response in the new host. We can also try and minimize the number of alloantigenic differences between the donor and the recipient. So, of course, blood type testing, also called ABO blood typing, is a way where we can minimize that. And certainly we can also eliminate the hyperacute phase of rejection patterns just by making sure that the patients have the right blood type. There's also an analogous procedure for tissue typing that's very similar in concept to blood typing, but we're just looking at the differences in the HLA alleles. Remember, HLA, human leukocyte antigens, is synonymous with MHCs. We can sort of see how many differences there are between a host and donor MHC, and we can try and reduce the number of differences because that correlates with increased chance of survival. Now, right now, we've identified 3 co-dominant loci. These are parts of genes that are primarily responsible for rejection. I won't say anything as large as the majority, but these have the largest determining factors of tissue success or graft rejection. These loci, the fact that they're co-dominant, are inherited by both parents, and which means that there are 6 possible combinations of these loci. So tissue typing today currently looks at the number of mismatches between these 3 co-dominant alleles. So you have a much higher percent chance of the graft surviving, as the graph on the right suggests, when there are 0 mismatches between these 6 alleles, as opposed to when there are all 6 mismatching from each other. Now, unlike blood typing tests, tissue typing does take time. We can do this for kidney transplants. Kidneys last 72 hours on ice, and patients themselves can undergo dialysis. It's a totally routine procedure. So patients waiting for a kidney transplant can afford to take the time to make sure the tissue typing brings back the lowest number of mismatched HLA alleles in order to ensure the highest chances of that graft survival. Heart transplant patients, however, cannot afford to take that time. Not only will a heart not last that long outside of a body, but the patients won't either. A heart-lung machine is nowhere near as great of an instrument as a dialysis. It itself has a lot of complications. And for that reason, HLA typing is not considered in pairing with potential donors. So in the case of heart transplants, it's really just ABO blood typing and anatomic compatibility that's considered when finding a match for the donor and the host. Things like the paucity of the heart donor, the emergent need for transplantation, the dire need of the situation itself, and also the recent successes of other immunosuppressive therapies override any benefit of reduced HLA mismatches between the donor and the recipient. So having said that, now we're going to talk about these immunosuppressive strategies. Now, the bulk of immunosuppressive strategies rely on factors that we've already learned previously in the activation of T and B cell chapters. So it's really taking that understanding and then figuring out, okay, how can we manipulate these in order to prevent activation in the cases where we don't want it, such as in this case where we want a successful graft to happen. So a lot of the first strategies that came about were means of inhibiting T cell signaling pathways. The first 2 major drugs that accomplished this were cyclosporine and tacrolimus, which themselves inhibit the genes that transcode cytokine like IL-2, thus ultimately blocking IL-2 synthesis. IL-2, of course, we learned is the T cell growth hormone, quote unquote. So T cells will not proliferate without IL-2. So before cyclosporine in particular, the majority of transplanted hearts and livers would ultimately fail. But just the simple introduction of this drug really switched the pathway and the success of these grafts. So just after that, the majority of these grafts survived more than 5 years. So a very significant breakthrough in the medical community. Another drug is rapamycin, which doesn't block the transcribing of IL-2, but it blocks the IL-2-driven proliferation by disrupting the intracellular mechanisms that get turned on once IL-2 binds. Also, downstream, what happens when the T cell receptor binds to its antigen and the CD28 co-stimulator pathways bind to their ligands, B7. What's kind of cool about rapamycin is that it notably does not impair survival of T regulatory cells that do need IL-2, or at least not nearly as much as non-regulatory T cells, because these Tregs are needed to promote immune suppression. So we want these, and we don't want the effective versions of T cells, obviously. Another drug that has nothing to do with the IL-2 pathway is mycophenolate myophotol, or MMF, which is simply an enzyme that blocks enzymatic activity that's necessary for both lymphocyte maturation, so this is before these lymphocytes actually ever mature, and then also even activated T lymphocytes that are fully mature and have been activated blocks the necessary enzymatic activity within the cells that's necessary for proliferation. Other strategies include monoclonal antibodies. What's kind of cool is that OKT3 was the very first approved monoclonal antibody that went through clinical trials. And this happened to target CD3, which is a well-known marker for T cells. And so by doing so, it would bind to T cells, thus activating complement, and activating phagocytes and natural killer cells through their Fc gamma receptors. Another one is anti-CD25. CD25 is what we know of as the alpha chain and on the IL-2R receptor. So it blocks IL-2 binding without inhibiting the signaling pathways like we just learned about. Another example, alemtuzumab, which is an anti-CD52 monoclonal antibody, targets both B cells and T cells. So CD52 is a little bit less understood, but it's found on all lymphocytes. So therefore, the antibody will bind, and just like with the anti-CD52, actively complement phagocytes and natural killer cells. We can also block activation indirectly by blocking the co-stimulatory pathways. So a very common drug is to take a CTLA4 and combine it with an IgG fragment. So this is a fusion drug that relies on the CTLA4 outcompeting for B7, thus blocking that stimulatory pathway. And then in addition to that, blocking CD40 by having antibodies specific to CD40L, thus preventing that ligand from binding to the receptor on the various innate effector functions in B cells. We also might want to get rid of the alloantibodies or the alloreactive B cells. We could do this through plasmapheresis, which is actually the removal of a patient's plasma while retaining the blood cells and circulating the blood cells back into the patients. So by removing the plasma, we also remove the alloreactive antibodies. We can also introduce intravenous IG therapy, where we introduce the donor's plasma that has the alloantibodies into the host patient. So the theory on this is that the Fc portion of these antibodies outcompete for the alloreactive antibodies, potentially taking the valuable competing space, for instance, for the Fc neonatal receptor, and thus these alloreactive antibodies get cleared a lot faster compared to the infused ones that you're pulling in from the donor patient. Lastly, instead of getting rid of the antibodies themselves, remove their source via B cell depletion, and we could do this through anti-CD20 monoclonal antibodies like rituximab that, again, bind to these B cells. CD20 is a very known marker for B cells, activate complement, and activate through the Fc gamma receptors, phagocytes, and natural killer cells. And lastly, an issue we haven't talked about too much in this particular class, but general anti-inflammatory drugs like corticosteroids that we hear about very often in sports injuries in order to reduce inflammation that can augment the adaptive immune response. Now, immunosuppressive strategies have dramatically and significantly improved graft survival in recent years. It used to be before the introduction of the very first blockbuster immunosuppressive drug, cyclosporin, that the 1-year survival rate of unrelated cadaveric grafts was around 50 to 60%, and grafts from living related donors was around 90%. After the introduction of cyclosporin, the increase in the unrelated cadaveric grafts increased to over 90%. It even brought a much more difficult graft, the heart transplant, up to 90% for a 1-year survival rate, the 5-year still being at 75%, a very significant increase from what used to be an almost guaranteed rejection. Now, these immunosuppressive strategies are typically introduced in sort of 2 phases. One would be an induction therapy, which is both done preoperatively and then immediately after transplantation, usually in much higher amounts and much more complex cocktails of these immunosuppressants. And then the patient is gradually weaned off those and shifted into a long-term maintenance strategy. And these combined strategies have all but eliminated both the hyper and the acute rejection phase and shifted all obstacles in our current line of research into combating and controlling immune reactions during chronic rejection patterns. Now, as you may have guessed, there are a lot of side effects associated with these immunosuppressive strategy. The first one being that we are suppressing the immune system. So the primary defense against viruses and other intracellular pathogen is severely compromised by these strategies. Latent viruses that typically lay dormant within our systems are particularly prone to pouncing upon this opportunity. So viruses like the herpes simplex virus or the Epstein-Barr virus are notable side reactions to immunosuppression. Patients are also particularly at a higher risk for cancer. Of course, CD8 cells are known for combating cancerous cells, and we'll get into the next chapter. But then we've suppressed that primary defense against that. Furthermore, many viruses are themselves responsible for inducing cancer. So not only have we directly eliminated the primary defense against cancer, but we've indirectly promoted cancer by suppressing the main defense mechanism against the viruses that themselves might cause cancer. Furthermore, there's higher risk with what we would call opportunistic infections, things like bacteria and fungi that we typically don't associate with normal diseases because our body's very well attuned to combating against it, and they only pop up in immunosuppressive patients. Now, despite these associated side effects, which we typically associate with the immune system, the major limitation of the tolerated doses of most of these drugs is the direct toxicity to cells unrelated to immune suppression whatsoever. So these drugs are themselves incredibly toxic. Cyclosporine is itself notably toxic to kidney functions, so it can result in the declining renal function of kidney allograft recipients in particular. Now, in the previous chapter, we learned about methods to induce tolerance to self-antigens. Here, the goal would be to induce tolerance and achieve a lack of an immune response to these foreign donor cells or donor grafts, even after withdrawing immunosuppressive drugs. We would like to achieve these through the same mechanisms through we achieve self-tolerance, namely anergy, deletion of cells that react to allogenic grafts, and then also suppression via T regulatory T cells. Now, the beauty of this is that tolerance will achieve all the benefits of immune suppression without any of the side effects. We could still allow the T cells, our own host T cells and other lymphocytes to react against other foreign antigens as they're intended to, or against cancer cells. Thus, we wouldn't be susceptible to increased infections, the development of tumors, and we certainly wouldn't be relying on these drugs that are themselves incredibly toxic as well. Now, we have a couple of different strategies that we hope to either promote this or achieve central tolerance. One is through a co-stimulator blockade. So the idea is that whenever we first transplant a graft, just due to the ischemic damage associated with the surgical procedure itself, that promote damage-associated molecular patterns and the recognition by the receptors that would thus recognize them. We have a lot of co-stimulators that usually we associate just self-antigens or allogenic MHCs not being reactive themselves. So because of the fact that we have co-stimulators, that produces the initial activation and thus the continual reaction against these grafts. So if we block these co-stimulators in the first place, then we have rather benign MHCs that even if they're not self, wouldn't activate a T cell, just like we would achieve in peripheral tolerance. The second strategy involves the transfer of hematopoietic stem cells, which would achieve hematopoietic chimerism. This is where our bodies would adopt the donor HSCs as long as having the host HSCs and produce leukocytes from both lineages, and thus inducing tolerance in the central stage as opposed to just the peripheral stage. Lastly, there is hope of transferring or inducing T regulatory cells that are specific to donor tissue. Again, T regulatory T cells, just like they do in a host, would suppress the action of T cells that are reactive against allogenic material. So we want to promote this type of T cell activity while suppressing the effector T cell activity against those particular antigens. Now we're going to switch paces a little bit, and we're going to talk about compatibility to blood transfusions, which is itself a type of graft or transplantation. What's different from this is that blood cells are themselves leukocytes, but we haven't talked about them the entire semester because they are themselves not an immune cell. They do not express MHCs, platelets. Red blood cells do not have nuclei, so they do not have class 1 nor class 2 MHCs, unlike every other antigen-presenting cell in the body. But they do cause transfusion reactions via a specific type of antigen called ABO antigens. They're a class of antigens. Now, because they don't have MHCs, they're not associated with T cells at all. They're associated entirely with antibodies. So antibodies mediate all compatibility with blood. These antibodies will activate complement, result in hemolysis of the blood cells, and potentially release toxic hemoglobin, which is the primary but not the entire cause of rejection in blood transfusions. Now, these ABO antigens are polymorphic carbohydrate antigens on the cell surface of these red blood cells. Individuals typically will have some combination of either the A or B antigen and be tolerant thus of the ones that they do have and intolerant of the ones that they lack. Their method of intolerance is that they have natural antibodies that are generated specifically against the other type that they lack. So for instance, a person with type A blood type has the type A antigens of the red blood cell, but they also have antibodies that react against the B antigen that they don't have. This is just like an individual that has type B blood. That means the red blood cells have type B antigens, but they also have the anti-A antigen IgM antibodies present that react against the A antigen. Individuals that have both types of antigens are thus tolerant to both antigens and have no antibodies that react against either. This means that because they have no antibodies that react against these antigens, they are considered universal recipients. They can accept blood of any type because they're not going to react against it. This is the exact opposite of individuals with type O blood that have neither antigen. Now, because they have neither antigen, they are not tolerant of either of these. They have antibodies that react against both, so they cannot accept any blood with any of these antigens. However, they can donate their blood, and anyone can accept type O blood because it doesn't have these antigens. So it doesn't matter if they're type A or type B. Those antigens are not present on type O blood. So thus, people with type O blood are considered universal donors. Now, to close off our lecture, I'm going to finish off with a very unique procedure involving the transplantation of hematopoietic stem cells, or HSCs. A hematopoietic cell transplantation is also referred to as a bone marrow transplantation because this is where the origin of all leukocytes occur. It's in the bone marrow, and that's where these HSCs reside, differentiate, and proliferate. This is used to treat a number of different clinical procedures involving lethal diseases caused by intrinsic defects in one or more hematopoietic lineages within a patient. It all starts in practice with the donor receiving an induction or infusion of colony-stimulating factor, which triggers the increased production of HSCs within their own bone marrow. Thus, whenever they remove it and transplant it, there's a nice healthy population of these HSCs. Meanwhile, the recipient, in preparation for this procedure, is given a combination or one of chemotherapy, immunotherapy, and irradiation, which is meant to completely eliminate almost all traces of their own HSCs. So at this point, they are completely immunocompromised. They have no HSCs, and then the transplantation from the donor occurs. And then these donated HSCs re-achieve the natural equilibria within the host. Right? We always have a certain amount of HSCs and T cells and memory T cells within our systems, and this is meant to re-achieve that. So the mechanism that partially achieves that is not only the external irradiation and eradication, maybe is a better word, of the host HSCs, but then also what we refer to as the graft versus host effect, where the allogenic, meaning the donor T cells that are transplanted, kill all the remaining disease cells. Those donor T cells, of course, are not reactive against the donor tissue or other HSCs, but they will react to the new host cells in the same way that this entire chapter we've talked about the reverse direction, where the host T cells react against the donor cells. Now it's the donor T cells react against the host cells. And we want this to a certain extent because this is in part how we're treating the disease. We want to eradicate all of the disease cells and replace it with the nice healthy cells. And we're relying on those healthy cells to in part achieve that. Now, in general, a lot of complications are associated with HSC transplantation. One is that these hematopoietic stem cells are often rejected by even the most immuno-incompetent hosts. That means even through irradiation and immunosuppressive drugs, the body still has a natural ability to fight against this. One additional complication that appears in HSC transplantation and not in other types of grafts is that offspring still tend to reject grafts from their parents, which tends to violate one of the central principles of graft rejection, in that because they are tolerant of some of the antigens present on the donated grafts from a parent, that they should be tolerant to these. But we don't see this as much in HSC transplantation. Obviously, there are a lot of mechanisms of HSC transplantation that are not well understood, but it is known that natural killer cells do play a larger role in bone marrow transplantation in comparison to other types of graft transplants. And it's hypothesized that natural killer cells won't recognize the MHC in the donor tissue. This is something we learned in previous chapters, that some viruses cunningly have learned ways of evading CTL responses by downregulating class 1 MHCs, but natural killer cells come as sort of like a backdoor preventative mechanism. If they fail to recognize class 1 MHCs, they are promoted into activating and thus killing cells whose MHCs they don't recognize. So this is a hypothesized mechanism for rejection in HSC transplantation. There are two additional and severe reactions or side effects to bone marrow transplantation, the one being what's referred to as graft versus host disease. This is almost the same as the graft versus host effect, but it's a description of the intended and chronic pathological consequences to the host when this really gets out of hand. and it's the principal cause of mortality in HSC transplantations or bone marrow transplantations. Now, what's primarily responsible for graft versus host diseases aren't MHCs because of the severity of this effect, MHCs are always carefully matched in order to give the right pairing and the least amount of mismatches in the HLA alleles, as we discussed earlier. So it's the minor histocompatibility complex that play a larger role in graft versus host disease. Now, unfortunately for us, these minor histocompatibility complexes are not as well understood as the typical major MHCs. Good news for you is that then we're not going to dedicate multiple chapters to learning about them. So, nice. One future possible strategy of preventing graft versus host disease is possible screening and filtering of both natural killer and T cells from the bone marrow so that hopefully we only transplant the HSCs and not all the other leukocytes that come with it. Another common side effect is just general immunosuppression. So successful transplantation often results in immunocompromised patients, meaning that we might possibly not reach that re-equilibria of natural leukocytes. And furthermore, even think of the preparative strategies, the radiation and chemotherapy. These are meant to kill all HSCs, but they of course also kill all the built-up memory T and B cells that are necessary for secondary and tertiary responses. And all of a sudden we don't have those anymore. It's almost like starting from a blank slate. So patients are very prone to infections after HSCs, increased susceptibility to cancer and opportunistic infections, as we discussed in general patients that are on immunosuppressive therapies. A few strategies in trying to achieve the effects of HSC transplantation, namely eradication of deficient diseases somewhere along the HSC lineage with nice healthy cells, is potentially the transplanting of not HSCs but embryonic stem cells, maybe induced pluripotent stem cells that we've taken from the host themselves, or even CRISPR-edited allogenic cells. These are all hypothesized methods of solutions to the graft versus host diseases. And lastly, I'm going to close out this chapter having just talked about graft versus host diseases and HSC transplantation with a very famous case from a bone marrow transplantation involving what's now known internationally as the Berlin patient. So Timothy Ray Brown from, you guessed it, Seattle, Washington, was a patient who had HIV, who actually had AIDS as a result of HIV, received hemopoietic stem cell transplants from two donors that had a very specific mutation of their cells with a lack of a functioning CCR5 receptor. This is called CCR5 delta 32, and then had to go off antiviral medication. So the purpose of the HSC transplantation was to treat leukemia. And of course, in order to do so, they gave him irradiation and chemotherapy, eradicated all of his own HSCs, and then he got this transplantation that eventually gave him HSC and leukocyte counts characteristic of the donors that also had this mutation. As a result, HIV counts plummeted because he had no functioning CD4 cells that were infected anymore. They were all eradicated, and through the graft versus host effect, were killed by the donor T cells. The new CD4 counts increased, the ones that didn't have HIV. And because of this, he was one of the first patients ever considered, quote unquote, cured, although the use of that term is debatable in the medical community. And he describes the process of going through this graft versus host disease as incredibly painful, incredibly bad, but it did result in the curing of this virus. Maybe a path forward is to somehow use this while mitigating the graft versus host disease and only taking advantage of the effect and potentially eradicating other novel types of diseases. So, as always, thanks for listening.